can think of PCR as molecular photocopying. You have one page of your DNA sequence or gene of interest, but it's too small for you to work on, so you're going to want to make more copies of it. But just how exactly does it work? Well, it's a little bit more complicated than photocopying. First, you need the 5 core ingredients: your DNA sequence of interest, primers to initiate DNA synthesis, DNA nucleotide bases or the building blocks, Taq polymerase to add these nucleotide bases, and a buffer solution to set the optimal conditions. Let's follow the journey of this little DNA sequence. Step 1 is called denaturation. Think of it as taking a swim in a warm, bubbling jacuzzi, but make it cute because it's actually nearly 100 degrees Celsius hot. It denatures our DNA sequence particularly by breaking the hydrogen bonds between the bases and causing it to separate into 2 single-stranded templates for replication. This lasts for about 15 seconds to 2 minutes to make sure that the strands have separated completely. Step 2 is called annealing. This time, think of it as going out of the hot jacuzzi, but you forgot your towel and it's a windy day, so all of a sudden you're shivering from the 40 to 60 degrees Celsius cold. This step allows the primers in your solution to bind or anneal to the denatured single strands, a forward primer for the 5' to 3' strand, and a reverse primer for the 3' to 5' strand. This lasts for about 15 to 60 seconds. Step 3 is called extending. It's too cold, so you'd rather extend your time by going back into the jacuzzi than going home. Why not? Jacuzzis are relaxing. So the temperature is increased again, but now only to around 70 degrees Celsius for 1 to 2 minutes. This allows the thermostable Taq polymerase to do its thing and synthesize new DNA. And that's it! You've made 2 copies of your DNA sequence. Really simple, right? Why not do it manually with test tubes and hot and cold water baths and saving money instead of buying a PCR machine then? Well, 2 copies isn't going to be enough for analysis. What we did is just 1 cycle, and you're probably going to need around 20 to 40 more. And thankfully, the PCR machine can just keep on going with these 3 basic steps again and again until you have enough copies of your DNA sequence. Now, remember, unlike a photocopy machine, you aren't making 1 additional copy per cycle. PCR machines amplify your DNA sequence exponentially. You begin with 2 copies in the first cycle, 4 copies in the second, then 8, 16, 32, and so on. So it's much more efficient than manually heating and cooling your DNA sequence, which would, not to mention, take forever to accomplish. PCR is a relatively fast and efficient method of amplifying small segments of a DNA sequence so that you have enough DNA for molecular or genetic analysis of fingerprints in a crime scene or detection of viruses like SARS-CoV-2, for example. And that's it. Thank you for listening.